Hi, good morning. This is an intimidating auditorium, but we'll move on. Today I'm going to talk to you about that sexy subject of sleep. Unfortunately, in the NICU, it's not so sexy. Before I start, I want you to keep in mind one interesting fact, actually two interesting facts. The infant's brain weighs 2% of the body weight, yet it is responsible for 20% of the energy intake in the body. It is pretty unrealistic for us to think that sleep is just restorative in nature and doesn't have any other major functions. What we do know now is that it is essential for growth and development, modulation of immune response, cognition, and physiological state. It has significant impact on all organ systems and is responsible for proliferation, neural development, brain plasticity, and overall network development. Calorie conservation and energy saving during sleep promotes feeding, weight gain, and ultimately improves the infant-parent dyad. Unfortunately, sleep is influenced by environmental factors, genetic factors, and physiological state. Coming from the environment of the uterus, floating in a warm bag of amniotic fluid, and listening to the drum of the maternal heartbeat, the premature infant is catapulted into NICU land. NICU land surrounded by bright lights, noise, painful procedures, frequent handling, which has potential detrimental effect on neurodevelopmental outcome. This study focuses on mid to late preterm infants. According to the World Health Organization, mid to late preterms are defined as infants born between 32 and 36 plus 6 weeks gestation. These infants account for 74% of preterm infants admitted to the neonatal unit, and unfortunately this number is rising. Unfortunately, these infants are the largely understudied infants in neonatal research. This is possibly due to the fact that the majority of these infants transgress through the neonatal unit, requiring minimal respiratory support, and their only concerns in general are feeding, weight gain, and maintaining temperature before they're discharged home. The objective of this study is just to take a snapshot of the sleep architecture of these infants while in the neonatal unit. I'm going to look at the sleep-wake cycles of these infants, and I'm going to quantify disturbances. Sleep-wake cycling is the alteration that occurs between states. It is evident on the EEG through the alteration in bandwidth, as sleep states can be identified as active, quiet, and indeterminate sleep. AEG, as can be seen here, is a processed, compressed portion of the raw EEG, which makes sleep-wake cycling quite easy to visible to neonatologists and to the nursing staff. Both AEG and EEG have a unique contribution to make towards sleep inside the neonatal unit, or detecting sleep. It must also be remembered that sleep is not static, and as the infant grows, there is alterations in how it is represented on the EEG. This is an example of an infant born at 35 weeks gestation, with the EEG recorded at 36 plus 3 weeks, corrected gestational age over an 8-hour period. As can be seen in the thickening of the band, this is an area of quiet sleep. It is represented by high amplitude bursts, followed by periods of quiescence and interburst intervals. This is another example of an EEG of the same infant, 35 plus 4 weeks gestation, over an 8-hour period of active sleep. You can see the narrowing of the band here, and it is represented on the raw EEG by high frequency, low amplitude waveforms. The study design. This study was carried out in the neonatal unit in COMH in Cork. It is a single-centred pilot study, observational study. The participants were clinically stable, mid to late preterm infants, and we studied 30 in this study. The procedures involved were overnight video EEG recorded prior to 36 weeks gestation or prior to discharge, whichever came first. What was reported was visual analysis of the AEG and the EEG, and we quantified the sleep-wake cycles and disturbances. This is an example. I'm going to show you 3 AEGs of 3 different infants. Each of these infants was born at 34 plus 5 weeks gestation, and their EEG pre-discharge was recorded at 36 weeks gestation. The weight of these infants varied between 2.2 kilos and 2.35 kilos. As you can see, I've identified on the AEG 4 periods of interruption: woke spontaneously, involuntary waking, or undisturbed nasogastric feeds, or how I have labelled these. I'm just showing you a brief look at 3 AEGs so that they can be viewed at for comparative purposes. You can see for AEG for infant 1, there was 4 sleep cycles identified. The green circles indicate the period of wakefulness and the period of disturbance. You can see a difference between A, B, and C, which showed us that between infants of the same gestation, there is differentiation between the amount of sleep-wake cycles. Unfortunately, as you can see on the slides, the amount of involuntary wakening is quite excessive. Infant C had all spontaneous wakings. You can see the rhythmic progression of the sleep-wake cycling in comparison to infant 1, who had all interrupted wakening. Okay, the results of this study, the clinical demographics, we did not discriminate with gender, and it's just the way they were recruited. 50% were male, 50% female. The mean gestational age in weeks was almost 35 weeks, with a birth weight of 2.12 kilos. Our correct gestational age of EEG was 35 plus 7 weeks. 20% of these were infants on phototherapy when EEG was recorded. 60% of infants were nursed in cots, while 40% in incubators. This is possibly attributed to the fact that half of those infants nursed in incubators were on phototherapy at the time of recording. The mode of feeding was all oral was 14, which was equivalent to 46%. Similar number were mixed feeding, while 10% of the infants were tube feeding only. When we looked at the sleep-wake cycles, the average number of sleep-wake cycles was 6 over the 12-hour period. Sleep interruptions were an average 2 with a range of 1 to 3, and these occurred during active sleep and quiet sleep, with the majority of the infants being woken or being interrupted during active sleep. The total awakenings overnight for this group of infants over a 12-hour period was 130 over 182, or 71% of awakenings spontaneously. However, the sad part is that 52 of these awakenings, or almost 30%, were interrupted. The number of sleep-wake cycles was found to be significantly higher in the incubator, babies nursing in an incubator versus a cot, although our numbers were quite small. No significant association was made between nasogastric feeds and the amount of sleep-wake cycles. So the take-home message from this talk is, it is disturbing that 30% of the awakenings in the NICU are actually involuntary. Sleep education is required in the NICU. This is not only required for doctors, nurses, but also perhaps for parents. Parents often interpret rapid eye movement as the infant about to wake for their sleep. Same with nurses and same with doctors. Unfortunately, further education would clarify that. So where are we going now with the sleepy study? Since submission of this abstract, we have recruited over 100 infants. They have all had overnight EEG monitoring, and we are following these infants up in the clinic. Thank you for listening, and I'll take any questions now.